Hi, everyone. First, I would like to thank the organizer to giving me the opportunity to talk today. And today, I would like to present an exciting project that aims at enhancing AAV gene therapy using a machine learning approach to overcome the packaging limitation in capsids. So, as some of you may know, gene therapy is a swiftly progressing field and holds promises to treat a broad range of diseases and not just limited to genetic conditions anymore. The main delivery vehicle for gene therapy are adeno-associated virus, or AAV, and these viruses can be re-engineered to deliver a therapeutical DNA into human cells. But yet, these treatments come with a hefty price tag, mostly attributable to the bespoke nature and the intricate technique required for delivery. The main limitation for AAV gene therapy are the payload limitation. The therapeutical gene or transgene they carry can't exceed 4.4 kb, which overrule all 6% of human gene and all gene editing system for being delivered. So to overcome this limitation, there's different strategies. And in our project, we are exploring one of them, which aims at reducing the space or the volume occupied by the DNA cargo. And so for that, we are developing a tool that is able to predict how a DNA fit into a AAV capsid. And this is a non-trivial task due to given the complexity and the lack of knowledge of the factors that influence the fit. We believe that the best choice of tool is machine learning due to its ability to learn from complex patterns and make some prediction on learned data. And so our tool will be able to take any DNA sequence, will be able to predict its secondary structure, will be able then to predict its tertiary structure, and then predict how well it fits into an AAV capsid. With the end goal will be to enhance the packaging efficiency, package more substantial transgene despite the payload limitation, and ultimately lead to a decrease of the cost of the treatment. And so one of the main challenges that we faced for this project is the absence of a pre-existing data set. There was no much data available that encapsulating the complexity of tertiary structure of DNA and an absence of data set of DNA back into an AAV capsid. So to tackle this challenge, we have created our own comprehensive data set using molecular dynamic simulation. This way we had a data set of diverse sequence lengths, of sequence with different degree of complexity. But for the molecular dynamic simulation, we couldn't include all the elements of the system because there would be, like the capsid actually, like you can see on the video, because it would be too large and complex to handle. And so the target for our machine learning model will be the compaction. And so we explore different metrics for that. We firstly look at the volume and then the radius of gyration. We take into account the mass distribution. But the volume can actually be misleading. For example, for two sequences of different degree of complexity, they actually can exhibit the similar volume. So we believe that the radius of gyration was actually more adequate for the task. And so also, when we explore the relationship between the radius of gyration and the length of the DNA sequence, as on these dot plots, we realize that a sequence of 3,000 nucleotides can have a lower radius of gyration than a sequence of 1,000 nucleotides. So this shows the complexity of the task, and for that, we believe machine learning is the best approach to harness the factors at play. And so after we have created our data set, the next step was to select a model for our machine learning. But the important consideration to have at first is that our data set is relatively small, and that is due to the fact that we are creating the data set ourselves, and each molecular dynamic simulation takes a lot of time to run. And so, given these constraints, we need a model that is able to train on small data set and a model that is resistant to overfitting. So we explore different models. We explore logistic regression, we did some support vector regression, and we did also random forest. At first, random forest seems to show promise with a high performance matrix, which shows quite a accurately prediction. But then when we dive into the learning curve, it shows an important caveat, which is that the model is actually overfitting. Whereas when we look at the random forest, that also shows some robust performance metrics, the learning curve shows a good generalization over the data and no overfitting at site. And further, when we dive into the QQ plot and the residual plot of the SVR model, it further substantiates the model reliability and assess its prediction accuracy. And so, at the heart of these machine learning initiatives lays the fundamental questions: what factors are crucial to predict how DNA sequence will fit into an AAV capsid? And so for that, our model classified the different features that we used. And you can see on this plot that the features classified as negative are actually detrimental for the prediction. And the features classified as positive are beneficial to make the prediction. And you can see that is not just one feature, but multiple features that contributes to make a prediction of how the DNA fits into the capsid. And so this advances our understanding of the factors that are crucial for the prediction and the fit, and enable us to make more accurate prediction, and ultimately lead to to tailor more efficient gene therapy treatments. And so our next step will be to evaluate our model by rigorous lab testing. And so for that, we're going to select different DNA sequence for which the model already predicts the degree of compaction. And then we will synthesize the DNA sequence, we will encapsulate them into AAV vectors, and then we will measure the yield of the sequence. Following the result of this evaluation, we whether want to confirm or infirm that our predictions are correct. If our predictions are incorrect, then we will go back to train our model. We will increase our data sets. We will change features, add features. We'll try different models until our predictions are accurate. And so to summarize, our project aims at addressing a bottleneck of the field, which is the packaging limitation for AAVs. And by leveraging cutting-edge machine learning, we've been able to unravel the different key factors that influence the fit, and we're able to develop a machine learning tool that predicts how random DNA sequence fits into a capsid. This journey has been challenging. At first, we didn't have a data set, so we had to create this data set ourselves using molecular dynamic simulations. And then we used this data set to train our model, machine learning model. And so the support vector regression comes to be the most promising model for the task, with a good generalization of the data, with no overfitting, and a high and good performance metrics. This model also helps us to understand what are the key features to predict how the DNA fit into a capsid, and this insight paving us the way to a more efficient and effective gene therapy treatments. And so our next step will be rigorous lab testing. We'll take our machine learning to the test and refine and iterate until our prediction aligns with reality. And so that's it for me. I would like to acknowledge my group, the Transformational Bioinformatics at CSIRO, Michael Cooper from Data 61, and our collaborators from the Children's Medical Research Institute. Thank you.
Thank you, Anne. Do we have any questions? Okay, I guess I'll start off. I was curious about which features did you find important?
Well, unfortunately, we are in the process of protecting our data in our workflow, so I can't. That's why I had to hide everything. Sorry. So yeah,
okay.
I can't say it.
Okay. And so, in creating your data set,
Yeah.
did you generate? So, did you extract features from the molecular dynamics project trajectory, or?
Well, we did extract features from the sequence, the structure, and the simulation.